Dr. Anirban, good morning and welcome to the webinar. Hello. Doctor, good morning. Dr. Biju, good morning. Dr. Sindhu, Dr. Natesh, and Dr. Sapna, all of you, good morning and welcome. We have with us Dr. Anirban, who is the director at NITTE University Center for Science Education and Research. And he is also the Dean of Faculty of Biological Sciences. Dr. Anirban will be speaking on the discovery of microRNA and its role in post-transcriptional gene regulation. That's the research that won Nobel Prize this year by Victor Ambrose and Gary Rufkin. This year's Nobel Prize honors these two scientists for their discovery of a fundamental principle governing how gene activity regulated. Dr. Victor Ambrose and Gary Rufkin were interested in how different cell types develop. They discovered microRNA, a new class of tiny RNA molecules that play a crucial role in gene regulation. Their groundbreaking discovery revealed a completely new principle of gene regulation that turned out to be essential for multicellular organisms, including humans. So Dr. Anirban will be speaking on their research on microRNAs. Now we have a brief CV of Dr. Anirban's research accomplishments. Before going to his talk, let me give a brief overview of it. Dr. Anirban is a Bachelor of Fisheries and Master of Fisheries from College of Fisheries, Mangalore, which is affiliated to the University of Agricultural Sciences, Bangalore. And later he pursued his PhD in Japan, and present designation, as I told, is the Director, NUCSER, and is also Dean of Faculty of Biological Sciences at the NITTE University. His expertise include cancer genetics and zebrafish biology, molecular biology, and biotechnology. He has published more than 86 research papers in national and international journals of repute, and Q1, Q2 journals 46. Cumulative impact factor is very high, 260.6, and H index Scopus 21. Citation is 1700, and he has supervised 5 PhD students for their research, and 5 more are undergoing guidance under his supervision. And gen bank submissions include 40, and he has 5 patents to his credit, and he is a very accomplished scientist. He is also CEO NITTE Government of Karnataka Center of Excellence in Aquamarine Innovation, and he is the founder of Consortium of Rare Genetic and Bone Marrow Disorders India Network at NITTE University. He is also the founder of the Cancer Gene Database at NITTE University. I'm very proud that I was also his college mate in College of Fisheries, Mangalore. With this very brief background, he has given only 3 lines of biodata. Thanks, Anirban, for saving time. And I am sure whatever I told is a very, very brief tip of the iceberg, I can say. You have accomplished much more than what I have told. This very brief background, I once again welcome Dr. Anirban. We have with us Dr. Biju, a national award winner in science communication, Dr. Sapna Anigol, national award winning teacher, Dr. Sindhu, Dr. Sanat Kumar, and Dr. Qureshi are all with us. Many seniors will be joining, or some have joined on YouTube also. We have both audience in both Cisco Webex and YouTube platform. For all of you, welcome. And now over to Dr. Anirban. Please take over and deliver your talk. Thanks. Thank you.
Dr. Kushal Das sir is also joined, sir.
Yeah, welcome, Dr. Kushal Das sir. He is the director at BLE University. He is also a very well-known scientist. Welcome to you, sir. So now over to you, Dr. Anirban, for your talk.
Right. First of all, thank you very much, sir, for considering me worthy of giving a presentation on a topic which is a Nobel Prize awardee this year. And it's an honor for me to be chosen for presenting this work of Victor Ambros and Gary Ruvkun, their work on microRNA. I thank KSTA profusely for giving me the opportunity, particularly to Ramesh sir, for this wonderful opportunity for presenting this, the seminal work that has been done by these two gentlemen. It's a big challenge, of course, to summarize work that was considered worthy of a Nobel Prize this year. However, what we have learned or what we know so far about microRNA, all these started from the discovery that these two gentlemen made and the kind of work that followed. And today we are a lot more aware of what microRNAs are, how are they produced, what they do, and what are the implications of this particular discovery. Since it is impossible to summarize everything that they have done, so what I have done here in my presentation is I'll try to give a very brief introduction to the concept at the beginning, and then try to explain some of the results, and of course give a brief into the developments that happened post the discovery of these microRNAs by these two extraordinary gentlemen. In the presentation, I have kept it very simple, very minimal, just to ensure that it is not becoming too technical. The idea is not to perhaps explain or discuss each result of the several papers that they have published, but to drive home the point what they published and what is the impact that their discovery has made so far and what is next that is going to come. So on that note, let me start with the presentation. I think, okay. So before we get into the scientific accomplishments, let's know the awardees a bit more. I'm sure some of you would have read about them. So did I. I do not know them personally, of course, but then I looked up. Primary source of information for me is the Nobel press release that was published on the database, on the website. So whatever I'm going to say here most of them are based on the information that is available on the press release, and some information that I've collected from various sources. I also would like to mention here that what I'm going to explain about microRNAs and their role in gene regulation is what I understood based on the publications that I have gone through or kind of work that I've done so far in my scientific career. If there are some clarifications to be made, I'm more than willing to clarify them at the end of the presentation. And I hope I'll be able to answer all the questions that are going to be put forward. So Victor Ambros and Gary Ruvkun, both of them actually started their journey together. And there is a great example of how collaboration between scientists help achieve something extraordinary. In this case, both of them started almost at the same time. They did their postdoc together, and then the good part was that they discussed their research before they published in the same journal, in the same year, in the same issue, just consecutive papers that came up in Cell. Now, Victor Ambrose, right now, he is a Silverman Professor of Natural Sciences at the University of Massachusetts in the US. And Gary Ruvkun is a Professor of Genetics at the Harvard Medical School. And he's also associated with the Massachusetts General Hospital in the same city. So a little bit about Victor Ambrose. Why I put this slide is for us to know how people start their journey from very humble beginnings and how they go on to become very important in the society and the contribution that they bring about in terms of for the benefit of the mankind. Now, Victor Ambrose he was born in a very village kind of a setup in a farmland in New Hampshire in 1953. Then he went on to do his PhD from MIT in 1975. And then he did his PhD in 1979. His boss, his guide was Professor Baltimore, who was also a Nobel laureate. And I don't think many people get this opportunity to be under the supervision of two Nobel laureates. So during his PhD, his guide was a Nobel laureate, and then he joined the lab of Robert Horwich, who was also a Nobel laureate, and he did his postdoc in his lab along with Gary Ruvkun. He was also at the postdoc in the same lab. And then he got into the faculty positions at Harvard from 1985 to 1992. He was with Harvard University, and then 1990 to 2001 he was with Dartmouth College. In fact, what his postdoc supervisor mentioned, that when he was not given a position in Harvard, he made a mention that you are potentially losing a Nobel laureate. I read it somewhere in the press release where it was mentioned that his postdoc guide was a little disappointed that he was not given a position there, and his prophecy came true. Eventually, at the age of 70, he was awarded the Nobel Prize this year. In 1993, during his postdoc, he published the first major paper which actually led to this discovery, and that was about this microRNA called Lin4. So, and as you all know, this discovery happened in Caenorhabditis elegans, which is a worm model that is used in biomedical research. And then, of course, as I mentioned, he was there with Harvard University for some time. And then later, he joined University of Massachusetts Medical School. And he's continuing there. He's holding the position of the professor. In case of Gary Ruvkun, his story is equally interesting. Victor Ambrose remained in the field of biology, started with biology, remained in biology, and went on to do molecular biology, and then looked into how genes are regulated in a simple organism called Caenorhabditis elegans. In case of Gary, he was born 1 year earlier to Victor. He was born in California in 1952. And interestingly, he did his BA in biophysics, which is not so closely connected to the traditional or conventional biology that we talk about. And he went on to do his PhD in biophysics at the Harvard University, which he completed in 1982. And then, as I mentioned, he, along with Victor Ambrose, joined the lab of Robert Horwich at MIT. And later, he did his second postdoc at Walter Gilbert's lab in the same institution for about 3 years after he completed his postdoc at Horwich's lab. And along with Victor, in 1993, he published a paper in the same journal called Cell, where he basically described the mechanism which by which the microRNA LIN4 controls the expression of its target protein or target mRNA. So the papers were consecutive in terms of their appearance in the journal. Victor Ambrose's paper came first, where it was mentioned what LIN4 is and how it is controlling the regulation of an mRNA called LIN14. You will get to hear these two words quite a lot in my presentation. So LIN4 is the non-coding gene and LIN14 is the coding gene or the coding mRNA. So he basically, the paper which Gary Ruvkun published described how the negative regulation of LIN14 is carried out by LIN4. And that was, of course, again done in C. elegans. And probably Ruvkun's major contribution to this discovery actually came in 2000, in the year 2000, where he discovered the second microRNA, which is called LET7. And actually that changed a lot of things in the scientific community. I'll come back to it later when I present this towards the end of the presentation. So LIN4 was the first microRNA that was discovered in C. elegans, and LET7 is the second one that was discovered by Gary Ruvkun. And the discovery of LET7 actually made things quite clear in the scientific community that these are indeed potent regulators, new regulators of gene expression in not only in C. elegans, but also in higher animals, in fact, across the evolution. And after his postdoc, he, of course, he continued at Harvard. He still continues to be there. He is a professor of genetics, and he is continuing his research on microRNA and gene regulations. Now, look at his story as a scientist who started with biophysics, went on to do something basic molecular biology, and then goes on to discover something so very important that plays such a critical role. Now, this is something that I collected from Nature Navigator, and this is the list of publications that started from the time he discovered LET7 microRNA, Ruvkun's second paper, which came in 2000. So this graph actually chronicles the total number of publications that started from 2002 till 2024. This data is 3 days old. So perhaps in 2024, we may have more publications right now. But nevertheless, what it shows is the impact of their discovery. Now, number of publications to date related to microRNA and gene regulation is about 61,589. And the number of researchers around the world working on this particular area is almost 2 lakh people. 1,97,718 researchers working in this field. And as you see, the graph is going up and up. And the good part is that microRNAs are still being discovered. It has been predicted that there are about 1,900 microRNAs present in human genome, but true microRNAs or genuine microRNAs are about 500 that has been discovered, but I'm sure there will be many more that are going to come in the future. So the major paper that is cited in the press release of Victor Ambrose and Gary Ruvkun's Nobel Prize, the first paper that is cited and the first paper that actually describes LIN4 is this paper, which came in 1993 in the December 3rd issue of Cell. And if you see the volume 17, I've just remembered the number 843 to 854, the page number. And Ruvkun's paper was from 855. So here he mentioned the complementarity of LIN4 with LIN14. So just remember this, I have highlighted this LIN4 and LIN14. remember these two keywords as we move into the presentation. Now, this paper has received total of more than 18,000 citations as on 7th of November 2024. So just imagine the kind of discovery that this paper brought in and the kind of impact that it has made. Similarly, the paper from Rupkun's lab, of course, there's a slight mistake on the top. That is, please read it as Rupkun's lab. So this is the same paper, same journal, same issue, same volume, consecutive article that came immediately after the Victor Ambrose's paper was published. And here, as I mentioned at the beginning, here Rupkun's team showed how Lin4 actually controls Lin14. So this is again in the same issue in 1993. So two papers coming from two scientists, and apparently when they published this paper, they had a discussion together and decided to publish upon the discussion that they had, and they wanted to know whether the other team, which is working on something similar, is actually getting the same kind of results or not. So that's kind of a discussion which was probably which is very relevant in today's context also, because sometimes as scientists we try to work in silos and we try to safeguard our results. We try to keep them without making them known to others. But sometimes what happens, if we do discuss, chances are that we end up getting benefited. And this is one great example where two competing scientists, they are working on the similar topic, yet came together, discussed everything, and then published. So now what are the salient features of these two gentlemen's discovery? So what they basically did, they discovered a non-coding RNA, and they found that that non-coding RNA has a role in gene regulation, basically control of gene expression. So basically, if you put these two together, so they discovered a new type of regulatory non-coding RNA. Now, to understand these two concepts of non-coding RNA and gene regulation, we'll have to go back into a little bit of basics, and the next few slides will probably give you some idea on what these are all about. And then we come back to the story of Gary Victor's and Gary's story of this microRNA, and then we summarize it and conclude it. So now this is called the central dogma of molecular biology, but there are exceptions to it. I will tell you about the exceptions at the next slide. But then what we already know is how genetic information flows in a cell. So when you say genetic information, it is the information that is defined in DNA and is converted into a functional product in the form of a protein. And we know that the DNA is transcribed into an RNA, and the RNA is translated into a protein. And we also know that all RNAs that are transcribed do not become proteins. So the ones that do not become proteins, the ones that become proteins are called coding RNAs, whereas the RNAs that do not become proteins, which basically means which do not undergo translation, and hence there is no protein of them, they are called non-coding RNAs. Now, there are many different types of non-coding RNAs. We know the classical conventional non-coding RNAs, which are most abundant. And in the next slide, I will just take you through very briefly. I'm sure many of you are already aware of this, but this I'm trying to build the story here so that it becomes easier for you to connect to what they discovered. Now, non-coding RNAs, if we divide them into two major types, one is called the housekeeping non-coding RNAs. The other one is regulatory non-coding RNAs. So Gary Rupkun and Victor Ambrose discovered a new class of regulatory non-coding RNA. So what are the housekeeping non-coding RNAs that we already know? The first one is tRNA. We already know what tRNAs are. Basically, they are the ones that bring the anticodon and the amino acid which is linked to that. ribosomal RNAs, which are very important for the formation of the ribosomes, SNO RNAs, which are small nucleolar RNAs that are meant for facilitating the processing of ribosomal RNA in the nucleus. And we also have small nuclear RNA or snRNA, which is meant for major processing of mRNA, splicing, etc. In the regulatory type, there are two major kinds. Of course, earlier it was thought, maybe till 5 years back, it was thought we only have linear non-coding RNA, but now we also have something called circular non-coding RNA. So the field is getting more and more complex as more and more things are getting discovered. Nevertheless, in the linear one, there are two different kinds again. One is that small non-coding RNA, which are less than 200 nucleotides in length. The other one is long non-coding RNA, which are more than 200 nucleotides in length. And if we focus only on the small non-coding RNA, which has less than 200 nucleotides in length, there are three major categories in that. The first one is microRNA, which is the topic of today's discussion. Then we have siRNA, small interfering RNA, which all of us are very aware of. In fact, this is very routinely used in molecular biology to knock down a particular gene and to know the function of a gene. Incidentally, microRNAs and siRNAs work almost in a similar manner. The biogenesis, the process that they undertake in becoming mature is also very identical. However, there are slight differences. I'll try to cover this at the end. And then there is the last one is spy RNA or PV interacting RNA. These are again double-stranded RNA molecules that are specifically interacting with PU proteins, which are expressed in the gonads, in the gonadal organs. Now, if you look at this, so the entire structure of these non-coding RNAs, there are so many different kinds. And then this microRNA is perhaps the most widely studied at this point of time for its role in gene regulation. Now, the next part that we need to speak about is what is gene regulation. Basically, it is a mechanism or the process that decides how much of a protein should be produced in a cell and at what stage each protein should be produced. When you say protein, basically they should be produced from the mRNA. So basically the control happens at the RNA level and at the protein level. And this kind of changes depending on various needs, developmental functioning, adaptation to environments, etc. And this control happens at various stages during the flow of the genetic information from DNA to RNA, from RNA to protein. So if we are trying to look at the control that is happening at the RNA stage, then we call it transcriptional, which is the control that happens before the transcription takes place. And then again, there is a control which happens after the transcription has happened, which is called post-transcription. And when we try to see the control at the protein level, the same thing you can imagine is that the control, there is a set of control which happens when the protein is getting produced. And then there is another set of control that happens when the protein has been produced. Once the protein is produced, how the control happens. So let me quickly go through these two major kinds of controls. The first one is, of course, transcriptional regulation. And the first player in transcriptional regulation is called transcription factors. Now, why all this information become necessary is just to have you an idea how much we understand when you talk about Lin4 and Lin14 story of Ambrose and Kuhn. So transcription factors basically are proteins. They are DNA binding proteins. They can bind to certain sequences in the DNA, and their binding allows the RNA polymerase, which is the major protein which is involved in the transcription of a gene, to make the gene transcription better. So of course, transcription factors can either help in increase gene expression, or it can help, or it can reduce expression of genes. So it is both kind of a on and off mechanism that we know about by these transcription factors. The next component that is important in regulation of gene at the transcription level is called promoters or enhancers. Basically, these are sequences of DNA which are present in the regulatory region of a gene. When you say regulatory region, the region which is upstream of the site where the transcription begins. So the promoters are generally very close to the transcription start site, whereas enhancers are a little far away from that site. So these are the sites, again, where the transcription factors come and bind. So the first is the protein that we talked about. Second is the sequence of the DNA, which allows the binding of that protein. So there has to be a perfect match there. The transcription factor should be able to find its target DNA, and then only it can bind there. So the role of promoters and enhancers are well understood in biology. We know that there are some techniques called enhancer trap assays, where we can use these enhancer sequence with reporter gene, and we can find basically we can find the entire set of genes that are responsible for driving the expression of a particular gene in a particular organ. So if one needs to know, say for example, if one needs to know what are the various genes that are responsible for development of heart, we can have an enhancer trap assay and find out all these genes that are based on this expression of a reporter line. So there are a lot of applications to that. The third one is actually quite important as well, which is known as epigenetic modifications. As the name suggests, modifying the bases without making any changes in the DNA sequence. And when you know modifying the bases, that basically means addition of certain chemical groups into either the DNA sequences or into proteins that are involved in determining which part of the gene is to be transcribed and which part should not be transcribed. So as we know, nucleosomes are basically the structures where the histone proteins where the part of a DNA is wrapped around histone proteins, and which forms like a string and the bead, which is necessary for compacting the entire chromatin into a very small size within the nucleus. So these histone proteins are some of the residues, amino acid residues in histone proteins are modified with certain functional groups. Most important one is perhaps acetylation, but then we have phosphorylation, ubiquitination, etc. And then when we come to the DNA level, we have certain sequences in the DNA, particularly in the promoter region, which are methylated, which means addition of certain methyl groups, which prevents the RNA polymerase to come and bind and helps in turn prevents the transcription of the gene. So when there is a need, this methyl group is removed, and then the chromatin structure is relaxed, and then these transcription factors are able to come and bind, and this transcription takes place. So there are enzymes that bring these methyl groups and binds, and there are some enzymes that remove these groups, so etc. So like this epigenetic regulation is believed to be imprinted. We are supposed to have a set of genes which is already even at birth, we are supposed to have that signature intact based on how it has been for the parents, and any change in this could potentially be problematic and could even result in susceptibility to certain conditions. And nowadays, a lot of focus is on cancer. Loss of epigenetic regulation is directly understood in cancer cases. And now, if we look at the post-transcriptional regulation, now this is what we talked about when the RNA is transcribed. And once the RNA is transcribed, then again, there are a set of regulations. The first one is RNA splicing. Of course, I talked about SNO RNAs, which are snRNAs, which are involved in this mechanism. So a lot of different proteins are involved in this. Basically, as we all know, splicing is removal of the non-coding part of the gene, which are the intronic regions, and then stitching all the coding parts of the gene to form the mature mRNA. And then, of course, capping the mRNA at the 5' end and addition of a poly A tail in the 3' end, making the full mRNA complete. Now, there is another one which is known as RNA editing. We all know about how it is done. The classical example is the presence of these apolipoprotein gene in liver and intestine. Basically, deamination is the process by which one nucleotide in this gene is changed from CAA to UAA, which results in formation of a truncated protein, and that truncated protein expresses in the intestine, whereas the full-length protein expresses in the liver. So this is done by a deaminase enzyme, which is present in the system. So depending on the need, there are many other examples, but perhaps this is the most classical example that we all know. And the third one is, of course, related to the topic that we're discussing today, miRNA or siRNA mediated regulation. So here, basically, these are small antisense molecules. They come in single strand and they go and bind to target mRNA. Now, miRNA basically targets mRNA through its 3' UTR, and the result is it does not allow it to be translated. Whereas siRNAs bind to the direct complementarity to the coding region of the mRNA, thereby leading or initiating the process of mRNA degradation or mRNA decay. So the process of the inhibition or the regulation is slightly different for miRNA and siRNA. miRNA basically brings about translation repression, whereas siRNA bring about endolytic cleavage or mRNA degradation. So the other aspect now that we are done with the transcription, we have talked about during the transcription and after transcription. Let's see what are the regulations that are in place in the cell during translation. So the first one is, of course, initiation factors, that we all know how these factors are necessary to bind to the 5' region of the mRNA, and how it allows the folding of the 3' UTR and binding to the forming the cap assembly and interacting with the ribosome, allowing the ribosome to scan through the mRNA, and so on. So this is initiation factor. These are, again, several proteins which are included there. And the second one is when the mRNA which is produced in excess, or when it is no longer required, or an mRNA which is defective or which is having some kind of anomalies, needs to be degraded. So there are many pathways in that. So basically, the mRNA decay is endonuclease dependent, or sometimes it is exonuclease dependent, or sometimes endonuclease dependent. So the first thing that happens is the poly A tail is removed, the cap in the 5' end is removed, and then depending on how the mRNA needs to be degraded, various enzymes, basically endonucleases and exonucleases, come and bind there. So it could be de-adenylation dependent, or it could be de-adenylation independent. So what I'm trying to focus here is that at each stage, the cell knows how to take care of the excess, how to take care of what something is not required. And this precise control is supposed to be intact in normal conditions. And we already know there are so many instances where in instances of pathogenic conditions, diseases, where there are some sort of problems in these basic control steps that we know about. And in the post-translational regulation, once the protein is produced, then there is, again, modifications of proteins. We all know how post-translational modifications of proteins are important. There is one small schematic that is shown here. Ubiquitination is perhaps the most common one, and then you also have methylation, acetylation, phosphorylation, sumolylation. So many of them are done, and some of them are irreversible, some of them are reversible, etc. So these chemical modifications affect the function of the protein. Sometimes they enhance the function, sometimes they reduce the function of the protein, sometimes they allow better localization within the cell, sometimes they help in better stability of the protein, etc. And then, of course, the most common part, the mechanism, as we saw in mRNA, proteins which are not properly folded, proteins which are not properly translated, proteins which are not completely formed in terms of its primary, secondary, tertiary structure. If something is not right, these are degraded. So the most common pathway is the proteasome pathway that you know about, which is carried out by these enzymes, which are basically a family of enzymes, which E1, E2, and E3 that we know about. Each one has its primary role to play. The final thing happens with the E3 enzyme, which is a ligase activity that attaches the ubiquitin molecule to the protein. And attachment of that molecule serves as a flag for that protein. And based on that flag, the proteasome machinery identifies this protein to be degraded, and it goes into the bin, and then the recycled products are come out, and then these are used by the cell separately. So now that we are already aware of what non-coding RNAs are and how gene regulation happens, let's go into the discovery of Ambrose and Ruvkun, and let's see. Pointer is a little problematic. So let's see what was already known before they made this discovery in 1993. So as I said, we are talking about these two important molecules. One is LIN4, which at that time was only called as a non-coding gene. The term microRNA came much later. So LIN4 is the non-coding aspect. So they're the crosstalk, what they understood or what they discovered between these two molecules. One is a coding molecule, the other one is a non-coding one. And the LIN4 is the non-coding one, whereas LIN14 is the coding molecule, which is basically a protein. So what was already known before they published this paper? So on the right-hand side, I've shown this life cycle of Caenorhabditis elegans. So basically, they have 4 larval stages, which last for about 3 days. And then once it is over, they go into the adult stage and they live for about 3 weeks. So these larval stages are called L1, L2, L3, L4. And because the development is extremely rapid, so there are a lot of changes that happen within just short span of time, but very rapidly in each one of them. So sometimes when the conditions are not favorable, they enter into a state of dormancy, kind of a state, which is called lower state, and they can live in that condition for several months without any food, without any development. And then it's kind of when the conditions become favorable again, they enter the cycle and become adults. So please see to that. So as you will probably understand, when the larval development is taking place, it is sequential. So L1, then L2, then L3, L4. So if that is the case, you can imagine that L1 is early stage of development and L4 would be the later stages of development in the larval development. So what was already known in before they published this paper is listed here. I'll go very slowly in this. So LIN4 is basically, they found that they developed a mutant C. elegans by LIN4 loss of function mutant. And they found that LIN4 is required for early stages of C. elegans development, and LIN4 is required at all the 4 stages. So basically what they found, if LIN4 is not there in C. elegans, the C. elegans display features of early stages act inappropriately or ectopically at later stages. Means the features which are supposed to be in L1 stage are seen in L2 or L3 or L4 stage in LIN4 loss of function mutants. So that means a growth retardation, you could say, happening in LIN4 mutants. On the other hand, what they found was the LIN14 mutants actually showed the reverse. The LIN14 mutants showed stages of late developmental stages inappropriately at early stages. So the features that a C. elegans larvae would display at L3 or L4 was already seen at L1 stage. So that means it was already old at a stage where a normal C. elegans would be not so old. So that means it was already clear that the phenotype between the phenotype of a Lin4 mutant and the phenotype of a Lin14 mutant are opposite to each other. So what they found based on this, what was identified was that Lin14 activity is required at early stage, and hence it expresses at higher level in L1 stage, and that specifies the programs or the pathways that are necessary at L1 stage. Whereas in case of later stages, lower levels of Lin14 needs to be maintained. So the higher level of Lin14 has to come down when the larvae moves to the later stage. So the Lin14 level has to come down as the larvae moves to the later stages for initiation of the late developmental stages. So that was clear. So they thought that, okay, there is some regulation happening there. And how they identified this regulation to be post-transcriptional was they looked at the level of Lin14 protein and compared this with the level of Lin14 mRNA. Now, as you know, the mRNA is a transcript level and Lin14 is the protein level, which is the next step after the mRNA is translated. So as I've shown here in the diagram here, you see in L1, if you look at this brown coloration as, say, representation of an expression of a protein, Lin14 highly brown at L1, but as it goes to L2, L3, L4, it is diminished, or this coloration is diminished. So that means the level of the protein is getting reduced as the larvae is moving from L1 to L2, L2 to L3, L3 to L4. Whereas in case of L14 mRNA, the expression is more or less constant. So what it indicated was to them that the mRNA is getting produced, just that the protein is not coming at a later stage. If the mRNA was not getting produced, then it would have been a transcriptional or post-transcriptional regulation. The fact that the mRNA is getting produced, that means the mRNA is there throughout, but then there is some way by which this mRNA is being blocked from translating into a protein. And that's why this protein is not getting produced at the later stages of larval development. And that gave a clue to them that there must be a negative regulation. Of course, by then they already knew that Lin4 is negatively regulated Lin14, and that regulation is post-transcription. So what did Ambrose and Ruvkun show in their Cell papers? So both of them worked on Lin4 and Lin14. I will go with what Ambrose showed first. So what they did before, with the background information that was already available before this 1993 paper was published. So first they cloned Lin4 by a technique called chromosome walking, which of course we can't describe between positive time, but anyway. So they cloned this product and then they analyzed the gene sequence and they confirmed that it does not code for a protein. So the ORF signals, etc., that you see in a coding gene was not present. So they were sure that this is getting transcribed as an RNA, but it is not getting translated as a protein. And then they also found that this Lin4 is actually giving two RNA transcripts, very small. One is of 22 nucleotides of length. The other one is 61 nucleotide in length. And when they analyzed the sequence of this transcript, they found that it has a complementarity to a repeated sequence in the 3' UTR, or the untranslated region of an mRNA called Lin14. So this is what described in the Cell paper by Ambrose's team. And then what Ruvkun's team found was that they were very sure that Lin14 downregulation was happening post-transcriptionally. By comparing the protein level and the RNA level, they found that RNA level is not getting diminished, but the protein is getting diminished at later stages. And this temporal regulation that they saw, for that to happen, Ambrose's team had already shown that Lin14 is the target of Lin4. So they found out that the 3' UTR of Lin14 is sufficient to regulate this mechanism. So not the other parts of the Lin14 gene, but only the 3' UTR of that gene was required for downregulation of this downregulation mechanism that they found. In addition, they found that this Lin4, sorry, it is not 34, Lin4 sequence is conserved among other species of Caenorhabditis elegans. So they looked at 2 other species and they found that other species of C. elegans also contain the same kind of sequence as Lin4 they found in elegans. And finally, they found that they looked at this sequence. They found that in the 3' UTR of Lin14, it's not 34, please read it as Lin14. So there is this nucleotide sequence which is CUCAGGA. This is a 9 nucleotide sequence which was repeated 7 times in the 3' UTR, and that has a complementarity. That means that is an antisense molecule or antisense sequence to a same 9 nucleotide sequence in the Lin4 RNA. So that means the Lin4 RNA sequence, if you see here, if you consider CUCAGGA, and if you consider C as the 5' end and A as the 3' end, and if you see the sequence which is written below, and if you consider U as the 3' end and G as the 5' end, you see there's a perfect complementarity of this sequence with the sequence written on top. So by these 9 major observations, Ambrose and Ruvkun showed that Lin4 is indeed a small RNA and it is not degrading the mRNA. It is preventing the translation of the mRNA into a protein. So this is the summary of that research that led to the discovery. I have already told this. So Lin4 is negatively regulating Lin14, and they looked at the phenotype and they found the phenotype was exactly opposite to each other. And then, as I told at the beginning, both these researchers discussed among themselves the result of their findings, and they came to the conclusion that this Lin4 is actually a microRNA which is binding to the 3' UTR of Lin14, and through this sequence similarity, of course, microRNA, as you know, it is a stem loop structure, so it has some nucleotides which are matching perfectly in that 3' UTR of Lin14. So from 1993 to 2000, what happened was a shift from skepticism to conviction. So when Lin4 was published by Ambrose was the first person who actually published Lin4, and then Ruvkun also worked on that same thing. From 93 to 2000, people were a little skeptical about this mechanism and they dismissed it, saying that it perhaps may be an unusual thing in a lower animal like C. elegans, and it may not be true for higher organisms like humans and other mammalian systems. And in 2000s, when Ruvkun's team published the second microRNA called LET7, that changed the perception. And that in that paper, Ruvkun showed that LET7 is not only present in almost all organisms across evolution, but it is conserved as well. The sequence identity is conserved. So that means this is perhaps a very important mechanism which is present in all species across evolution. And in 2001, he coined the term microRNA. So what are the developments that we witnessed after this seminal discovery of Ambrose and Rufkun. First thing that came up was the concept of RNAi. I'll try to briefly talk about it. Of course, RNAi separately was given a Nobel Prize in 2006 to Andrew and Craig Simello. They got that Nobel Prize in 2006. Subsequent researchers worked on the structure of the microRNA, how it forms, how it is synthesized, and many different microRNAs were identified. People finally understood how microRNAs are processed in the cell, how they generated, and once they are formed, what they do. And also, it became very clear that a single microRNA can target many mRNAs, and at the same time, a single gene can be regulated by multiple microRNAs. So this was proved, or these were the things that came up after this discovery of microRNA by Ambrose and Rufkun. So RNA interference, as I said, it is a mechanism which is present in plants, animals, humans. In fact, very similar to what we see in microRNA biogenesis. So basically, primarily, it is plants use it as a defense against viral infections, and even in microbes as well. So the machinery that is followed in the cell is very similar as microRNA follows in a cell. So the only difference is here, it is double-stranded RNA that is employed, which gets processed into single-stranded RNA, and then they go on to do their job. MicroRNAs are also like that. They are produced as a duplex, and then it gets processed in the cell, and then finally goes on to bind to the target mRNA. So the structure of a microRNA, of course, there are many stages. Based on the stage at which the microRNA is identified, there are different names to it. The first one is when it is just transcribed as a big long sequence with complementarity between its sequence. It forms in a kind of a hair loop structure that forms. Of course, it is synthesized by RNA polymerase II, the same polymerase that synthesizes all our mRNAs. So it also gets a 5' cap assembly and a 3' poly A tailing that comes in. So at that stage, they are called primary microRNA or pri-miRNA. And then these undergo splicing by certain enzymes, and then they form what is known as precursor microRNA. And then once this precursor microRNA is formed, it again undergoes splicing, it undergoes processing, and then the loop structure is removed, and then it becomes the mature microRNA, which is about 21 to 23 nucleotides. So now, unlike siRNA, which shows perfect complementarity with the target mRNA, microRNAs do not show a perfect complementarity. Among these 20 to 25 nucleotides that you have, about 2 to 7, about 6 to 8 nucleotides need to be perfectly matching. And that sequence is called the seed sequence. Of course, we also have a wobble theory there, but then microRNA, for it to be controlling a particular mRNA, or rather an mRNA to be a legitimate target of microRNA, the match at the seed sequence is very critical. If the seed sequence is not matching, then perhaps it is not a real target. It could be a pseudo target of a microRNA. So the rest of the sequence may have complementarity, may not have a complementarity with the 3' UTR, other parts of the 3' UTR, but that seed sequence, which is about 6 to 8 bases, that becomes very critical in microRNA. So the biogenesis that happens, the first thing, of course, it gets synthesized because it is transcribed as any other RNA. So the transcription happens in the nucleus. The first step is transcription by RNA polymerase II in the form of primary microRNAs. And these are processed or kind of chopped by these enzyme called DROSHA, which is present inside the nucleus. Now that chops it into smaller fragments, and these smaller fragments are called precursor microRNAs. And these precursor microRNAs are brought out from the nucleus. They are exported into the cytoplasm by a gene called exportin, by a protein called exportin. It's a kind of carrier protein. It brings it into the cytoplasm. And then in the cytoplasm, there is again another RNase 3 enzyme called dicer that cleaves the precursor microRNA or pre-microRNA one more time, and it removes that loop structure. And once that loop structure is removed, it gives a double-stranded structure called miRNA duplex. And this double-strand structure gets unwinded. One of them becomes an active strand, and the other one gets degraded, or the other strand, which is called the passenger strand, gets degraded. And there is no kind of rule that which one becomes active or which one becomes a passenger strand. Any of these two strands can become an active strand. And then the last part is loading of this strand into an assembly of protein, which is called RISC assembly. The RISC stands for RNA-induced silencing complex, which is a complex of several proteins. The most important protein in that complex is called Argonaute protein. And that takes this entire complex and takes it to the 3' UTR, finds its complementary sequence in the 3' UTR of the gene, and through its seed sequence identifies it and binds there. And then because of the binding of microRNA in the 3' UTR, the 3' folding of the 3' UTR along with the 5' UTR during the translation process does not take place, and hence ribosome cannot process this mRNA. The net result is translation repression. So as I said, microRNA and siRNA, all of us are using siRNAs quite extensively. We know there are a lot of similarities because they follow the same pathway, the same kind of enzymes digest these RNAs, non-coding RNAs. The mechanism is they are synthesized in double strands, and then they are processed as single-stranded RNAs. siRNAs either produced or either from external source. When you can put it, suppose in cell line experiments, we use double-stranded siRNA, and we expect the cell to process it, and then it goes and binds to the target site. And generally, when you try to look at the function of a certain gene, we design an siRNA against the gene, and then the siRNA, we expect it to bind 100% complementarity. So the binding should be perfect. Whereas in microRNA, as I said, the binding is not perfect, except that seed sequence, the rest of the part may bind or may not bind. But in siRNA, all the 21 to 23 nucleotides that we have is 100% match. It has to match 100%. So the differences that we see in these two non-coding RNAs, which are almost similar, is that, of course, siRNAs are derived as double-stranded at the beginning itself, whereas miRNAs are formed in the form of a hairpin structure, the loop kind of a structure from folded RNA transcripts. And the second point I already mentioned, it has a partial complementarity, whereas siRNAs have complete or full complementarity. MicroRNAs usually targets many mRNAs, primarily because it has partial complementarity. So the seed sequence, that short sequence of 6 to 7 nucleotides could be there in many UTRs, whereas generally siRNAs usually have a single target. MicroRNAs control gene expression by translation repression, whereas siRNAs ensure that the mRNA is degraded. And the length, I already said, they are 19 to 25 nucleotides, whereas siRNAs are 21 to 23. And primarily in the body, miRNAs are meant to control the expression of endogenous mRNA to perhaps allow a temporal or spatiotemporal expression of these mRNAs, because they don't want to degrade that mRNA. The mRNA remains in the cell, but whenever it is required, it is translated. When it is not required, it is repressed, as shown in the experiment by Ambrose and Ruvkun. L14 mRNA is present in L1, L2, L3, L4, but L14 protein is present only in L1. Whereas in L2, L3, and L4, it is repressed. Whereas siRNA basically is usually meant for as a defense mechanism, and it usually targets exogenous mRNA, particularly the ones that are derived from the pathogens. And if you look at clinical applications, so miRNAs are perhaps mainly as drug targets, quite extensively used in diagnostics and as biomarkers, whereas siRNA is basically meant for therapeutic purpose to prevent the synthesis of particular mRNA. So to conclude, what Rufkun and Gary Rufkun and Victor Ambrose found are these tiny RNAs with profound physiological relevance. They are meant for the proper miRNA function is necessary for normal development, important in normal physiology. And of course, there are so many examples now where we know that miRNAs are deregulated in many conditions, genetic conditions, including cancer. So what they understood was that mRNA is necessary. Without a normal mRNA function, development will be impacted. And then if there is a deregulation in the mRNA function or abnormal miRNA activity, either very high activity or very low activity, can be contributing to certain disease conditions. So this small RNA that they discovered turned out to be a very seminal discovery because it was unexpected. Second, it was not accepted for about 7 years. And then finally, the scientific community accepted it. And then what it did, it revealed a new dimension to gene regulation, and which is now quite convincingly accepted as something which is essential for all complex life forms. So with this, I end my presentation here. I'll be happy to take questions if there are any, and if I'm in a position to answer them. Thank you very much.
Dr. Anirban, super, very nice presentation from basics. You took us to the advanced research in case of mRNA, microRNA, and the works of Victor Ambrose and Gary Rufkun were very well explained by you. On behalf of KSTA and all the attendees who have joined both on Cisco WebEx and YouTube, we are immensely thankful to you for great presentation and also your time. Now we have with us senior professors. First, Professor Kushal Das sir is there with us. Sir, over to you for your comments. Sir, please unmute. Unmute. Hello. Kushal Das sir.
Unable to hear you, sir.
Yeah. Muted. We are not.
It is unmuted, but we are unable to hear you, sir. Please check the connectivity of the speaker.
Now we have with us Dr. Biju Dharmapalan also, Dr. Sindhu, Dr. Sanat Kumar, K.V. Prakash. All of you can comment on or give your.
Am I audible now?
Appreciation to Dr. Anirban. Yes,
Yes,
sir.
sir.
Yes, sir.
Am I audible? Okay, thank you. Good afternoon, sir.
Good afternoon, sir. Good afternoon.
Professor Chakraborthy, it was brilliant and extremely good lecture. Very
Thank you so much.
thoughtful, and is so beautifully explained the expression process of the both of you and the functional aspect, comparing with the microRNA with the siRNA. That's a really interesting part of the whole topic. We never expected that we'll bring in this two-way and comparison to make us better understanding. I'm Professor Kushal Das. I'm Distinguished Professor of Physiology of the B.M. Patil Medical College, Bijapur, and the BLD University.
Thank you so much. Thanks for your wonderful comments.
Thank you very much.
I'm glad you found it informative. Thank you so much.
Yes. And so for that, I must thank to you for the nice presentation, and thank to Dr. Ramesh, the CEO of KSTA, for making such an arrangement for your talk. It is actually, he made the whole series itself are very interesting. Yes. And as a medical person, that I do not able to have many things as details and depth as you have, and both the Nobel laureates, their works in that level. I have only one question, as I'm currently having some works, and me and my students
Yes, sir.
on the microRNA, basically I'm in vascular physiology, so on the vascular system.
Sure.
I have one question that the assessment and this thing, will it be only restricted to microRNA when we're supposed to find out a protein expression pattern and defect, specifically vascular disease and angiogenesis process, certain disease when we target and we use it in our laboratory. So we are only now selected only for microRNA, like our microRNA 385, like that. So do you think that any specific way for this particular gene expression protein expressions to understand thoroughly that why it is underexpressed or why it is not changed, even though microRNA change, do you think that siRNA should be simultaneously to be evaluated and assessed?
Yes, sir.
This is particularly in the therapeutic and disease concern for target proteins.
So if the application is for a therapeutic purpose, I would recommend an siRNA approach would be better, because for the simple reason that siRNA is a lot more specific than microRNAs. One of the impediments in microRNA based research is to find out the real target. So when we do an algorithmic search for potential targets of microRNA, we end up with a big list because it predicts based on the complementarity of the seed sequence.
Right.
Now, to prove which are the pseudo targets and which is the real target, it requires a lot of functional studies, which needs additional validation through many other methods which are available right now. But the fact still remains that one microRNA can potentially target many mRNAs, and at the same time, one mRNA can be targeted by many microRNAs. So the specificity part needs to be very well ascertained. In case of LIN4 and LIN14 mutants, they found something which was like opposite to each other. There was a clear kind of a jigsaw fitting there.
Right.
So the ones in LIN4 mutants were retarded, but the LIN14 mutants were already developed at a stage where they're not supposed to be developed. So, and then they did a bit of rescue experiment, and they found that actually it was able to rescue with a mutant LIN4, but not with a wild type LIN4, etc. So what I'm trying to tell you is that if the application is therapeutic, definitely siRNA would be a better approach.
Yeah, that's what I'm saying. That
for the reasons that I mentioned. But if the application is for a diagnostic purpose,
okay,
then, and if you're trying to evaluate the presence of a biomarker over a period of time, or whether if it is can be linked to the progression or the response of the patient to a particular treatment, then microRNA could be a good indicator to tell you whether there is a kind of a treatment happening or the treatment is working fine. And if it is working, and if that particular protein that the microRNA is, or that mRNA that the microRNA is supposed to target is primarily responsible for that pathological condition, that if the mRNA level is coming down, you should see the microRNA level going up. So it is always an inverse relationship. If mRNA is going up and the microRNA level is coming down, there is a chance that it is a proper target of that mRNA. So this is what is my understanding of the whole thing. I may be wrong, but this is what I feel is.
No, I think that there is a justification, because this is a question, because when we made it, these ones, we target the protein specifically to see that whether expression pattern changes and altered in a certain type of induced vascular diseases and all.
Correct.
So we want to also see certain molecules and drug target molecules, whether it acts or not. So in the corrected this or rectified, or there is no change in all of it. So for that reason, I was interested to know whether siRNA also we required to study or not. Anyway, you have given a good positive answer response, and a lot of thanks to you once again. Thank you very much, Dr. Ramesh, for giving me an opportunity. Thank you very much, sir.
Thank you, sir.
Thank you very much, sir. We have senior faculty, Dr. Sanat Kumar, and my friend KV Prakash, Dr. Biju Dharmapalan. Now the floor is open for you for your comments and appreciations.
Thank you, Anirban. As a
Hello, sir.
common man, I'm not qualified enough to make any statements on or give any opinion on that, but only the basic things like why C elegans is used and why connecting to Nobel laureates through simple approach, and that the impact of using this for future complex life forms. So in that context, thanks to KSTA and thank you, Dr. Anirban. Over to the experts to take it forward.
Thank you, sir.
Thank you.
Thank you, sir. Thank you.
Dr. Sanat Kumar. Umesh, you can ask questions that friends have asked.
There are few questions in the chat box as well as in the YouTube channel. Sir, there is first question by Dr. Pamisha. Protein, can they produce using artificial intelligence?
What is the question?
Using artificial intelligence, production of proteins can be done, sir?
Production of proteins using artificial intelligence. I don't think so it is possible because artificial intelligence works on training and memory. So basically, it is an intelligence which is manifested by a machine which needs to be trained for a particular aspect to be remembered and to predict when the same thing is perhaps given more and more period of time. So protein production is a fundamental biological process and it has to follow the central dogma that we know about. And it has to be, the genetic information has to flow from one stage to another and finally forms a protein. Now we can develop certain techniques using that protein, and maybe the most common thing would be like looking at a protein, interaction of a protein with some another protein. And we can train a machine through AI to give a signal every time that protein meets another protein, a signal is produced and AI should record that signal. So if we train that AI, you saying that every time the protein is going to target another protein, you have to record it, so the machine can record it. But I don't think a protein can be produced by an AI, no.
Thank you very much, sir. If epigenetics says the environment is important in gene expression, what are the environmental factors that influence gene expression, sir?
Oh, the gene expression, the most important environmental factor is the presence of favorable conditions in the environment. So when you say favorable condition, for a cell to divide, it needs two major things. One is the nutrient, other one is the availability of oxygen. So these are considered as most favorable conditions. So if a cell, because when a cell divides, there is a lot of energy that is being spent because there are so many things that need to be done. At the end, whatever it is produced should be equally divided to two daughter cells from one single cell, whatever is produced. And that division has to be absolutely equal. So there is a lot of energy that cell needs to spend. So when the condition is not favorable, the cell would not spend so much of energy getting into its reserve. So obviously favorable condition, environmental condition, it means presence of nutrients and oxygen for the cell. Now there is another way of looking at it. So when you talk about the expression of certain proteins or genes across developmental stages, something that is required at early stages of development may not be required at a later stage. In the same way, some cells divide rapidly and they die and they generate again very rapidly. Take, for example, red blood cells. We lose red blood cells in loads every single day and they divide so many times. But if you take a nerve cell, it grows only once and it doesn't divide. So there are certain requirements and these things are controlled by a set of pathways which are sometimes epigenetically controlled, sometimes controlled by many other mechanisms. One of them is microRNA mediated control, perhaps. So these are so many examples are there. Yeah.
Thank you, sir. Now, Mr. Ali Jawad, please unmute yourself and you can ask the question directly to sir.
Uh, Dear Dr. Chakraborty,
Yeah,
thank you so much for the informative session.
thank you.
I'm a PhD scholar working on microRNAs on hepatocellular carcinoma.
Okay.
So my question is, how can we pharmacologically target microRNAs, and should our primary focus be on microRNA biogenesis factors such as DROSA, dicer, or transcription factors to
Mhm.
effectively modulate microRNA's activity for therapeutic purposes?
So for that, the very simple thing is, if you are looking at, say, if you want to overexpress a particular microRNA, first of all, you need to see what is happening to that particular microRNA in the condition that you're looking at. Suppose a particular microRNA is targeting an oncoprotein, and that oncoprotein is driving hepatocellular carcinoma. So for some reason, that microRNA is not functional, so the oncoprotein is not getting inhibited. So you need to find out what is wrong in that pathway, whether there is any mutation in the genes that are responsible or that are playing a role in the biogenesis of the microRNA. It could be a mutation in the DROSA gene. It could be a mutation in the gene that codes for dicer. It could be a mutation in exportin. So many proteins are involved in the pathway anyway. So, or maybe Argonaute protein, or maybe any other members of the RISC assembly. So you need to find that first and see what is the difference that is happening. Now, if you need to look at, say, pharmacologically targeting a particular microRNA or any components in the microRNA pathway, one could perhaps think of forming certain nanovesicles, for instance, which can take a microRNA to a particular delivery site, release it there, and allow the microRNA to bind better to the RNA. Or it could be that make some changes in the, using CRISPR, make some changes in the microRNA binding site in the 3' UTR, which renders it getting detected by the microRNA. So as I said, a microRNA can target its mRNA through this 3' UTR complementarity. So if that complementarity is lost due to certain changes, the microRNA will no longer be able to bind, even if the microRNA is getting produced in the cell. So if that mRNA happens to be an oncoprotein, you can engineer it. You can, using site-directed mutagenesis, one can change it and make that microRNA bind to it again, and then make this microRNA work better. So there are many different ways one can target pharmacologically. You can have mimics, MIR mimics, which are available, which actually do the same job as microRNAs. But then oncomirs, there are mimics which are already available. Quite a few of them are there. So there are many different ways. Chemical compounds which mimic the function of microRNAs. That can also be done. Yeah.
Thank you, sir. Thank you for the answer.
Thank you, sir. Shri Vijay TS, please unmute yourself and you can ask the question.
Hi, Anirban sir. Thank you for this wonderful session. I just want to know, there's a lot of emphasis on protein intake has been, given by everyone nowadays. But how's the external protein what I'm consuming? How's that quantity and quality would affect the internal process?
Sorry, can you ask me the question again? I couldn't quite get it. Yeah.
Yes, sir. Yeah. There's a lot of emphasis given on protein intake nowadays as food. So I want to know how exactly the external protein food what I consume will affect my internal protein synthesis and everything. Whether it's the quantity or it's the quality of protein, how it is, because as I can see, this is, see, for a body which is already built for an adult body, it's already all there. I mean, how my external protein would affect these processes?
Well, I mean, when someone is prescribed certain proteins to be taken in, perhaps there is a fundamental problem in the body, which probably the protein metabolism is not all that good, or the body is simply not able to produce enough proteins. Now, what proteins are you talking about? If these are proteins which are meant for muscular growth, or if the proteins meant for intellectual ability, protein meant for better agility, protein meant for better hormonal control. There are many different things that need to be looked into. So we cannot just generalize it. When a protein is required, for example, the simplest example could be someone working out in the gym and they require high levels of proteins because they need their muscles to grow better. Now, whatever food intake that is happening is probably not able to supplement that much of requirement of the body because you are spending a lot of energy and your protein demand is more in the body. So someone would probably take extra proteins, which are probably compensating for the deficiency that the body is unable to make. Similarly, some could be, so there are plenty of examples of metabolic disorders, inborn errors of metabolism, where certain amino acids are not being synthesized, certain vitamins are not synthesized in the body, and these things are required to be supplemented from outside the body. How it goes and enters the body, it is the standard pathway that we know for anything that we take from the food. It goes into the gut, gets into the lumen, gets absorbed into the bloodstream, and through the bloodstream goes to the target site and does its job as whatever we are talking about, whatever conditions that we want to address to. So, so the protein requirement needs to be ascertained first. And if the protein is really required for the body, and if the body is unable to synthesize a good amount of protein, then of course that is to be done. Of course, there are certain conditions where people are, there are certain genetic conditions. One of them is something that I work with, where the ribosome itself is defective. The protein synthesis machinery in the body, which is supposed to produce all proteins in the cells, the ribosome itself is defective. So they are called ribosomopathies. So they end up with some anomalies. Primarily they have bone marrow failure, but they also have propensity to developing cancer, etc. So there are so many examples like that. So if your question is to know how external protein enters the body, it is the same pathway, same mechanism by which a protein which is synthesized in the body goes and does its job. I hope I have answered your question.
Yeah,
Thank
sir,
you very much, sir.
Actually,
Yeah. Anybody else? Dr. Madhu, Dr. Qureshi, Dr. Sindhu, Dr. Sanat Kumar, any of you can make comment or ask question if you have, otherwise we will close. Umesh, are there any more questions?
As of now, we have finished all the questions.
Yeah,
Right.
Thank you, sir.
Dr. Anirban, thank you very much for a wonderful presentation. Indeed, we are very lucky to have you for this presentation. You took us from the basics to advanced research that Nobel laureates have carried out. On behalf of KSTA and all those who have joined on Cisco WebEx and YouTube platform, once again, a big thanks to you for your time and wonderful presentation.
Thank you so much. Thank you, sir, for the opportunity. It was a pleasure interacting with all of you, with all the luminaries and the senior professors in the audience. I hope I have just done justice to the topic that was given to me. And I hope I could explain it the way the Nobel laureates would have done. But then it is always an honor. I hope someday I'll be able to meet them and maybe explain or share the story of this presentation if I ever get a chance to meet them. And I should also thank you, Ramesh sir, for making this happen. So all thanks primarily to you for actually thinking of me and extending this invitation to me. I look forward to be associated with KSTA in the future as well. And whatever you have in mind, if you think I'm worthy of it, please don't hesitate to tell me. I will do my best to live up to the expectations and also to deliver it according to or in the right way. Okay. Thank you so much, sir. Thanks for the opportunity.
Yeah, thank you, Dr. Anirban. We wish that you meet the Nobel laureates and also in future you'll be one among them.
Oh my God, that's too flattering, sir. But that's a heavy weight on my shoulder. But I don't know. But yeah, I keep saying this in all my presentations. Not everybody can win a Nobel Prize, but certainly we can all do a Nobel research. So I think that is primarily important. We should not, we should bring in some amount of novelty in everything that we do. Not necessarily that would end up with a Nobel Prize, but then we can always aspire for it. Yeah, no doubt about it.
All the best. God bless you. And I'm also thankful to Dr. Kushal Das sir, who
Thank you,
has been with us for all these 4 webinar series. And also senior professors are here, Dr. Sindhu, Dr. Sanat Kumar, my friend Biju Dharmapalan, National Award winner, and Dr. Sapna, Dr. Qureshi, Dr. Madhu, Dr. Maheshwarappa, Dr. Vijay. For all of you, a big thanks. Also, thanks to all the attendees, both on Cisco WebEx and YouTube platform. Thank you very much.
Thank you, sir. Thank you. I'll take your leave, sir.
Yeah, please. Thank you.
Thank you. Thank you.
Thank you.